Welcome to Digital PCR Decoded, where we unpack precision molecular analysis and its strategic implications. In this presentation, we'll explore how digital PCR works, why it changes the rules for absolute quantification, and where it can make the biggest impact in research and clinical settings, guiding you from core concepts to applied workflows and real-world use cases. This slide contrasts qPCR and digital PCR in practical terms. qPCR relies on threshold cycles and calibration curves, which can be biased by inhibitors and struggle at low copy numbers, whereas digital PCR partitions samples and performs direct counting to give absolute molecule counts, improved resistance to inhibitors, and much higher precision for scarce targets. At the heart of digital PCR are three principles. First, partitioning the sample into thousands of tiny reaction chambers to isolate single molecules. Second, applying Poisson statistics to correct for multiple targets per partition. And third, ensuring sufficient partition density so low abundance templates are reliably detected and quantified with minimal sampling error. The amplification and detection process in digital PCR is straightforward. Standard PCR chemistry runs independently inside each partition. Reactions proceed to endpoint saturation without continuous monitoring, and a binary fluorescence readout separates positive from negative partitions, simplifying interpretation and tolerating less than perfect amplification efficiencies while still producing absolute counts. Here we follow the workflow from imaging to validated quantification. High-speed scanners capture images of every partition. Automated software classifies bright versus dark partitions. Poisson correction converts positive fractions into absolute concentrations, and confidence intervals are generated to define limits of detection and quantification for robust assay validation. The technical workflow and clinical application slide shows how microfluidic droplet generation, surfactant-stabilized monodispersity, and scalable formats like 96-well processing come together to support high-throughput, reliable analysis. Importantly, some workflows eliminate library prep, enabling rapid oncology mutation detection that speeds clinical adoption and decision-making. Chip-based digital PCR uses fixed picoliter wells to simplify fluidics and reduce reagent use, enabling precise partitioning on silicon or plastic substrates. This approach integrates easily with automated loading and fluorescence staining, delivering exceptional sensitivity, lower sample input needs, and a streamlined workflow compatible with high-precision applications. Hydrogel bead encapsulation creates uniform, crystal-like arrays of bead-bound reactions that combine on-bead amplification with direct sequencing coupling, allowing amplified targets to be routed straight into sequencing flow cells. This architecture supports single-cell mutation analysis and precise library quantification, making it attractive for NGS workflows where ordered spatial arrays improve throughput and consistency. When applied to clinical monitoring, digital PCR delivers ultra-sensitive ctDNA detection, enabling identification of rare mutant alleles below typical qPCR thresholds, tracks treatment response and emergence of resistance in real time, accurately assesses copy number variations for therapy selection, and uses absolute counts to normalize longitudinal samples across time points. In clinical virology, digital PCR provides absolute quantification for targets like HIV-1 RNA, HBV DNA, and SARS-CoV-2 RNA, improving sensitivity in low-copy samples and enabling consistent cross-lab comparisons. By eliminating dependence on standard curves and reducing amplification bias, it yields robust detection in complex clinical matrices and early variant identification. Advanced multiplexing expands the market for digital PCR by combining high-capacity, multicolor fluorescence, and barcoded probes with nanoliter droplet systems that integrate real-time kinetics and endpoint digital counts. This hybrid approach increases target throughput and opens doors to broader genomics and transcriptomics, profiling markets driven by higher multiplex capacity. Finally, digital PCR can be adapted for low-resource, field-deployable diagnostics through battery-powered microfluidic chips, smartphone-based fluorescence readers, and lyophilized reagents. The operational flow is simple. Collect a field sample, run rapid on-site dPCR with a portable reader, and get immediate, accurate results useful for infectious disease surveillance, environmental testing, and decentralized personalized medicine.